So first question, and probably to you, Takako, what's next planned in the GemJ whole genome aggregation program?
Okay, thank you for the questioning. And so the Japan whole genome aggregation is made from the deposit data set, mainly from Biobank Japan 3000 and Tohoku Medical Megabank cohort of 5,000 whole genome sequence data. Again, this is the deposit data. And so we once complete the reanalysis of the deposit data and nothing left right now. So we are waiting for the next big deposition from the big genome sequence data analysis project. And once the data will be deposited, so we'll be planning to restart again the whole genome aggregation analysis for the Japanese population. That's it.
Great, thank you. And a question to you, Oliver. Oh, it's disappeared. But we'll go to the second question. Is Australian Genomics set up for bringing code to the data, the federated analysis approach coming from the cloud workstream and now being tested by GEL?
Should I field this, Tiff?
Yes.
If you wish, yeah, sure.
Sure. We are currently not using the endpoints that the cloud workstream has been developing, tests or OS endpoints. I think our first contribution towards is DIRS, so basically making data available directly from the warehouse. This goes back to a little bit to the federated setup that I mentioned earlier, where each flagship driver project for the most part has their own clinical sequencing environment and analytical pipelines. As the infrastructure improves, we will start making REST endpoints available, at least for data harmonization, but that's a little bit further out, I suspect.
Thank you. So Soichi, a question for you. How frequently the prospective cohort phenotype will be updated to the human phenotype ontology? And can you give an example within your cohort?
Okay, thank you for the question. So right now, so in Japan, so we use the phenotype, human phenotype ontology in the rare disease field. So but we are planning to use the HPO in the cohort study. So mainly focusing on the common diseases. So and for example, the Peter Robinson, Professor Peter Robinson published a paper about the capturing the blood test values in the HPO. So we would like to capture the we like to code the blood test by using the HPO code. So and we in the prospective cohort study, we have so many blood test data and the by environment data. So we are planning to code the blood test values in HPO code. So I have no ideas for the frequency, but if we receive the data, in this case, we would like to convert to the HPO, I think. So it's okay?
Thank you. Tiff, a couple of questions have come up around duo codes in the context of dynamic consent. And in addition to the question on the screen, a related question is that the machine readable consent poster or talk from yesterday referred to control and also to the use of duo-derived consent clauses. Can you tell us, Oliver also alluded to it in his talk, but, and I know that this has also had a lot of input from the Global Alliance perspective. Could you give us a bit of an overview from that perspective?
No, very happy to. So control offers participants granular secondary data use options that map directly through to duo codes. with plain language descriptions. And as Jonathan mentioned in the jury breakout earlier, this was presented at the last plenary in Boston. So in parallel to that work that Australian Genomics has been conducting with control, the jury ruse collaboration around the machine readable consent development was sort of underway. So the projects are sort of indirectly related, if you like. We were very much involved in the development of the machine readable consents, and it was informed by our experience from control.
Okay, thank you. Oliver, a question around data storage. How do you manage data at rest versus requested data?
We don't.
S3 versus Glacier. So perhaps you'd like to first tell me what S3 versus Glacier is.
So the difference is basically how much of our data is in active storage and is available on short notice at any time, and how much did we file away in a cabinet that takes a little bit longer to pull out and make available. Right now, pretty much all of our data is on active storage and available right away. That is partially possible because a lot of the flagships are on exome sequencing and the data is quite manageable. As the whole genome sequencing efforts are ramping up and are contributing, that probably does not become viable. We keep data active while patients are still being reviewed and curated, and then the data gets pushed to Glacier and becomes available within 48 hours.
Thank you. A question for GEM Japan, and I think this would go to you, Soichi. What approvals or permissions are required to browse and download the patient clinical data in the catalogue? Soichi or Takako? You'll need to unmute Soichi. Yep.
Okay, so in the case, okay, at first I would like to answer the question. So in the case of the Biobank Network, Biobank cross-search system, we provide the clinical data. So to choose the, to find the suitable biospecimen data. So, and the user can browse the clinical data in the web, on the web browsers, and they can download the clinical data in the Phenol packets. So, and to start use of our web service, so we ask the users to sign up the data use terms. So, and the data use terms defines how to say the restrictions of the clinical restrictions of the use of the clinical data. So the Biobank cross-search system is the purpose of the Biobank cross-search system is to find the suitable biospecimen data. So that's so if the user would like to use the clinical data or Phenol packet for the other purposes, it's not how to say allowed. So it's the restrictions for in the case of the Biobank cross-search system. So Takako, do you have any how to say additional answer?
Okay, thank you. And in general, the permission to use a patient clinical data is almost the same as the European countries on the globe. So principally, the data use, patient clinical data use is allowed within the range of informed consent of the participant or donors or patient. And the use can be approved by the Data Access Committee and the IRB of the institution of the user.
Great, thank you. A related question around consent to you, Tiffany. Does control also offer participants choice relating to which results they receive?
Yes, the initial rollout of control did have questions regarding the results that they're likely to receive on the basis of additional findings, for example, incidental findings. But this has also been informed by our current work looking at standard practices around sort of incidental findings associated with sort of research outcomes. And we're likely to update that work to reflect sort of the policies we're implementing now in Australian Genomics along those lines.
Great, thanks, Tiff. A question to you, Oliver. Has the has the process of getting labs to submit data to an S3 warehouse helped in the consolidation or understanding of sample metadata and provenance across different labs?
I think I would go with future tense on this one. Yes, we're learning certainly a lot, and how to manage the diversity of solutions in there. But it does help by talking to many different labs to understand what the key concepts are that every lab is tackling, and how to focus on those. So we're basically trying to come up almost with like a minimal set that we can extract consistently across all participating sites.
And a question to both you, Oliver, and Tiffany is, do you intend, or how do you intend to share data outside of Australia? And perhaps it might be a good idea, Oliver, to touch on the pilot project with Genomics England.
Sure. From the technical side, this is probably trivial. It's more a case of the policy and legal frameworks. Tiffany, do you want to touch on that one?
Tiff, you're muted.
Sorry about that. So from the sort of policy perspective, we certainly do lead our participants through a conversation around international data use in the consent conversation. Those that do provide consent for sharing internationally, that information, the data will be available via EGA where appropriate, by direct submission to international studies through our sort of data access committee process. But aggregate information will also be available through Variant Atlas that Oliver touched on earlier.
And in terms of technical parts, we can make data available either directly, hopefully in the near future through HTSCAT, but it's probably currently through pilot studies like the one that Catherine mentioned with Genomics England, where we're trying to figure out how does this work and how do we optimize the actual technical processes, which is a little bit easier in a one-to-one setting first before you're opening this up then to everyone else.
Right, thank you. And I think a great broad question that I'm going to ask you to address first, Takako, and then Tiffany, is where do you see the push coming from within Japan in terms of applying global alliance standards? And do you think, and what do you think the likely uptake of those standards will be?
Okay, thank you for asking. And so in GemJapan project, for example, for genome aggregation project, we use VCF, BAM, not yet CRAM, sorry, but it's a CRAM is under consideration. And another example will be presented by Soichi in the Biobank Network. Could you please introduce after me, Soichi?
Yes.
Yes, so, okay. Okay, so I already presented in my presentation, so how to say, yeah, so, yeah, so, anyway, so I'm sorry, I don't have anything to add, so.
That's absolutely fine.
Yeah.
And Tiff, from the Australian perspective around the push to apply data standards, would you like to make a general comment from the Australian genomics perspective, but then looking at the broader landscape within Australia?
Sure, Catherine. I think there is considerable interest in standardising approaches to genomic and health data capture and management. And I think not only within sort of Australian genomics, but between large genomic research programs, and then looking at more clinical trial applications and sort of other health system applications. And from the Australian genomics perspective, I guess our involvement in GA4GH has been so valuable because in contributing to the development of standards, we can ensure that they are fit for purpose to apply in Australia. And as a driver project, we're certainly very keen to promote and support implementation of GA4GH standards moving forward. And
I think just to make a comment at the higher level here is as Oliver mentioned, our government has made a large investment in genomics going forward. And so from the perspective of the roadmap and implementation plan over the next 8 to 10 years, we've really ensured in every iteration of that the engagement with the global community and learning from and application of the GA4GH standards is what we consider to be essential to the rollout of our sort of overall genomics initiatives. And I think that's certainly become part of the implementation plan that's currently being signed off by our health minister. So it is that issue of starting and demonstrating as I think we all have the value of the application of the standards, but then it really is important at a government engagement level that is recognised and it's seen as an ongoing process, because there's this continual generation of new standards. And I think, you know, in the breakout session, in terms of FASP and the connectivity between the standards, I think is incredibly important. And the goals are to continually add to that to make it easier and easier. And, you know, just from our perspective, to the example that Oliver gave around the acute care genomics, being able to start that project in the middle of this year and pull off the shelf all of the different standards and how we could link those together to making it as fast and efficient as possible has been incredibly important. So
Catherine, can I make a little bit comment on this issue?
Of course you can.
Thank you very much for giving me a time. So a little bit about some kind of broad perspective from Japanese side as well. So the data sharing is becoming a big issue also in Japan or Japanese research field. So currently, the promotion of medical research has been a major issue, duty of AMED so far. But now, data sharing of the research result, the sharing of the research result is becoming another big duty of AMED right now. And also, Japanese government has a big interest in data sharing, sharing a research result or reuse or produced data from our research. Because of the research budget is not so large, so we have to efficiently reuse the data to promote the research development on medical field in Japan. Thank you very much.
Great, thank you. And I think it's I think both examples from Australia and Japan are demonstrating that importance of getting that engagement at a government level as well, because that's the way that we can really roll it out. And then when we talked about GIF, it's then how do we start to look at sharing not just our experience, but our expertise and ultimately the access to data and the use of data internationally, because I think we all know you need the data on hundreds of thousands of millions of people from diverse ethnic backgrounds to be able to most accurately predict for the individual when it comes to its use, whether it's diagnosing a rare disease or looking at the pathogenicity of a particular cancer mutation. And then ultimately, as we move forward to look at pharmacogenomic variants and the application there, and we had a great section on pharmacogenomics in the GIF meeting. And then, we talked a bit about GWAS earlier today, but the use of polygenic risk scores for the prediction of common disease as well, let alone the applications at a population level around reproductive carrier screening, newborn screening. These are all of the issues that we're all going to be facing over the next 5 to 10 years as we're increasingly using genomics in clinical practice. So I think that's a great note to wrap up on as we're coming up to the hour. I'd just love to thank all the presenters and panelists, Oliver, Takako, Soichi and Tiffany. It was what's been a very rich session, and I think nothing brings the significance of the work of the Global Alliance to light more than seeing its application in the real world settings and seeing how it's making our lives so much easier. So thank you for all of our audience. That I'm very proud of you all. You've all stayed to the end. I think this has really been a fabulous session, and I'd like to thank you all for attending.